All right, guys, today we are going to be discussing bryophytes, lycophytes, and ferns. These are all spore-bearing plants. We're going to start with bryophytes, which are non-vascular plants, meaning they do not have vascular tissue, and we'll talk about more what that means in a moment. Then we'll go into our lycophytes, which do have vascular tissue, however, they still have spores, no seeds yet. And then our Monilophytes, which are our rest of our ferns, these guys have vascular tissue and true leaves. So your alternation of generations, which you should have completed for your pre-lab homework and uploaded to your Lab 3 submission on Canvas, this is kind of a review here, so this is what you should have written. I usually like to start with the spores section, so we're going to start there. But remember, spores go through mitosis in order to grow larger into a gametophyte. That gametophyte always produces gametes, right? Examples of gametes would be sperm and egg cells. They're going to fuse together and then create a zygote. Notice how we're starting with the haploid generation above, and we go into a diploid generation here. If you recall from meiosis, whenever you're going through meiosis in order to produce haploid structures like gametes, gametes are haploid, right? So you have one end here, one end here. They're going to fuse together and you get a diploid or a 2N zygote, right? In order to grow larger, again, that zygote is going to go through mitosis and it's going to grow into a sporophyte. Now remember, sporophytes always produce spores, but in this case, notice how we're going from a diploid generation into a haploid generation, and the only cellular division process that produces haploid cells is meiosis. So notice here, we are going through meiosis in order to produce haploid spores. So don't forget, we have diploid generation on the bottom and haploid generation on the top. So this is a very important process. This is the alternation of generations. Every single plant division is going to experience this in slightly more complex ways. So this is the base of the cycle, just at the very, basic way to look at it. So algae are going to do this. Bryophytes, lycophytes, and ferns will do this. Next week, we're going to talk about our seed-bearing plants. They're going to do this, but it's much more complex. So this is it's just very important to get this basic understanding first. So don't forget, gametophytes make gametes in what are called gametangia. So gametangia are little houses for the gametes, right? Think later on next week when we talk about pollen. Next week, pollen would be the example of the gametangia, and the gamete would be sperm, okay? So that kind of helps you put it into perspective a little bit. But we're not at pollen yet. We have not gotten there. We're only at our spore-bearing plants. So these guys do not have pollen grains. They only have spores. So gametophytes make gametes and gametangia. And then our sporophytes are going to make spores in houses called sporangia. So the sporangia house the spores. Okay, so don't forget that. All right, so this is our kingdom plantae. We're going to start, notice here we have this group, this division called Viridophytes. So these Viridophytes are anything with chlorophyll, right? So anything that is photosynthetic. So these two groups down here, these are actually our algae from last week. But we're going to get into what are called embryophytes, which are land plants. So these embryophytes include things like bryophytes, lycophytes, and ferns, our monilophytes. And then our seed-bearing plants we'll talk about next week. These are our spermatophytes. So embryophytes in general, these are our land plants. So this includes spore-bearing plants and seed plants. They are photosynthetic organisms. They might have pigments like chlorophyll A, chlorophyll B. They could have flavonoids or carotenoids. Notice how plants are green for the most part, right? Sometimes they do have other colors. And those other colors are attributed to the other pigments that they have present. Okay, so chlorophyll is green, and therefore these plants will reflect green light, right? And then the other pigments are reflected, resulting in those colors, right? So starch is our food reserve, okay? Cellulose provides structural cell wall support in plants, and we have this great land adaptation called a waxy cuticle. Okay, so the cuticle has this waxy covering on the epidermis or skin top layer of the leaves, and it just helps prevent drying out in plants, also known as desiccation. So the taxa that are boxed in red, you are going to be responsible for memorizing these and spelling them correctly. I won't take off very many points if you spell them incorrectly on the lab practical, but you have, your notes and everything available, so I will expect you to spell them correctly. So these are embryophytes, right? They all have embryos, so that is going to be at the base of your phylogenetic tree. When you're completing lab exercises, notice that page 28 has a phylogenetic tree just like this one. You're going to need to fill in these characteristics on each of the nodes, these inner node spaces here. So we're going to start with our bryophytes, which are our liverworts and mosses. Then we'll go into our lycophytes. Notice this word here, tracheophytes. We'll get into that more later, but that just means there's vascular tissue present. So our lycophytes we're going to talk about are lycopodiales and selaginales. Then we get into our ferns, which are monilophytes. These are xylitales and equisitales, as well as polypodiales. Okay. Now, this is incorrect. I just noticed it says seeds here. Polypodiales has spores. It does not have seeds. So this should be up here, really, on Spermatophyta. So do not mark that. Okay. So our first division that we're going to talk about is Marcantiophyta. So these are our liverworts. Some important things to know about them. Back to our alternation of generation cycle. Our gametophyte in this division is dominant. So in this division, our leafy green kind of part is going to be our gametophyte, okay? In liverworts, we describe, these aren't true leaves, and we describe this structure as thallus, okay? So they're not real leaves, are in ranks of 2 or 3, so you can kind of see them branch out a little in groups of 2, 1, 2, kind of like that. It's hard to tell from this photo. They also have specialized cells called oil bodies. These provide protection against cold temperatures or drying out, even herbivores from eating them, things like that. So our reproduction in our Marcantiophyta, our liverworts, so they require water, very similar to algae. They require water and they're photosynthetic. Those are two characteristics similar to algae in both our bryophytes and our Marcantiophyta. So the sperm actually need the water in order to swim to the egg. Our sperm here are flagellated. So they're going to swim to the egg, which is housed in the female gametophyte. We have this word here called archegonium. So the archegonium is the gametangia, or the house for the eggs. So there, once the sperm gets to the egg inside of this structure, they're going to fuse together, right, and create a diploid zygote, which grows into an embryo. That embryo is going to turn into a diploid sporophyte, already diploid. The embryo is already diploid, but it's going to turn into a sporophyte and then grow out of the gametophyte's archegonium, okay? So it is dependent on that female structure. So when it gets there, it's going to grow that whole new leafy thallus, and everything is going to grow right out of the female structure. These guys are also asexual. They can reproduce in what are called gemma cups. So these are little splash cups. So when a rain droplet comes along, it'll splash the required structures in order for fertilization to occur. So here is a really good picture of both the male and female parts in a Marcantiophyta. So in a liverwort, we can see on the left the female gametophyte, so our leafy green thallus down here, and we have our female gametangia at the top. This is called the archegonial head. So just remember that the archegonia houses the eggs, okay? So what's gonna happen, just like I mentioned before, those sperm cells, which are produced in the antheridial head on a male liverwort, sperm cells are produced up there. They're going to swim into the archegonium via water, right? Because they're flagellated, they're gonna swim up in there. Fertilization will happen. We're going to grow into a mature sporophyte and then produce spores all over again, okay? So those spores would germinate. We don't have true roots. We have what are called rhizoids. So they're going to germinate and then grow little, root-like structures, and then they can do the process all over again. So here you can see the female gametophyte a little better. Again, this is the whole thing up here is the archegonium. Eggs are housed down here. This is the male gametophyte, the antheridium, okay, with the sperm inside of it, all right? So this is what you will need to draw, okay, on your lab exercises. So this is more of a mushroom shape. This is what the archegonia looks like, cross section under the microscope. Those are the egg cells down here. Ignore number 4 and 2 and 5. Ignore those. Ignore 1. You just need to label that these are the egg cells right under here, okay? And remember, egg cells and the archegonium itself, these are all haploid structures, okay? These are not diploid structures. These are haploid structures. Same with the liverwort antheridia, the male gametangia. So here we have antheridial head, inside these sperm deposits. Again, haploid structures here. So our next division we're going to talk about is Bryophyta. So our Bryophyta, just like the overarching division name Bryophytes, so it gets kind of confusing. Bryophytes can be from the division Bryophyta as well as Marcantiophyta, okay? So liverworts and mosses are bryophytes, but this group, this division Bryophyta, is separate from Marcantiophyta. So these are little fuzzy carpet, mosses that you're used to seeing. Similar to the Marcantiophyta, these are also gametophyte dominant. So you will commonly see these in their little grassy moss stage. This is the alternative stage, so the sporophyte coming out at the top. You won't see that as often. We don't have true leaves, not yet. They are only 1 cell thick, okay? Leaves are arranged spirally, so you can see how they kind of whirl around the stem-like structure here. We do have rhizoids, so we don't have any true roots yet, just root-like structures called rhizoids. We have leaves that do exhibit, or leaf-like structures that do exhibit capillary action, so some water retained in there. And then we still have flagellated sperm that requires water to reach that egg house, right? So it requires water to reach the egg house, the archegonium. So when we get to our reproduction, the sperm, again, same situation. It's going to swim to the egg, which is located inside that archegonium. I highly recommend making a Quizlet for all these terms. So egg is inside the archegonium, going to fuse together with the sperm, create a diploid zygote, which grows into an embryo, right? That embryo is going to grow up into a sporophyte, which produces spores, right? And that sporophyte is growing directly out of the female house, right? Still. So notice here, you can see this even better in the mosses than you can in the liverworts. So this is our grassy green gametophyte at the bottom. We have male and female gametophytes, okay? But our gametophytes at the bottom, we are growing that sporophyte out of the gametophyte. Look at that. It's coming out at the top. Then that sporophyte, this structure from here to here, okay, is going to produce spores at the top inside of this little capsule. So the spores are produced up there. We have what's called an operculum, a little cap. That operculum is going to pop off once it desiccates and release spores everywhere. And remember, we're going through meiosis in order to produce spores. So those spores are all haploid every time. So here you can see this is going to be drawing number 10, the bryophyte life cycle. So this is our alternation of generations, but for bryophytes. So again, I like to start with the spores. Those spores are going to germinate into gametophytes. We have female and male gametophytes. They're going to separately, right, produce sperm and egg cells. The sperm, like I mentioned before, we have a rain event or some sort of water is introduced. Sperm's going to swim to those egg cells, fertilize the egg cell. Then we get a diploid zygote, grows through mitosis into a diploid embryo, still diploid, everything down here above, below the line, right, is all diploid now. So embryo through mitosis is going to grow into a sporophyte, right? The sporophyte's growing directly out of the archegonium. This is the female house here. So right down here where it's labeled foot, that's the base of the sporophyte. Okay, so that's where it's growing out of the archegonium here. So this new structure, that's the sporophyte. It's going to produce spores through meiosis, right? Those spores are haploid structures. They're going to dehisce or, spread, disperse everywhere. And then this whole thing happens all over again. Okay. So don't forget everything above the line, haploid, everything below the line, diploid. You're also going to need to draw the capsule of a sporophyte, of a moss. So inside of here, you would have spores inside of this capsule. Over here, you have these little babies of the moss, right? So the spores are growing into a gametophyte, right, after a while, but this is just kind of the starting point right when the spores are germinating. And again, you're going to need to draw those structures. So moss archegonia really up close. You can see archegonial head here. Now these are different. These are the moss archegonia and antheridia. They look a little different from the liverworts. Okay, so in moss, you have the deposits right here. So this is egg cell. These are egg cell deposits. In the antheridial head for moss, you have more of a, you can tell the sperm cell deposits are much larger. The egg cells are very small, okay? So kind of to get your mind situated here, we started with bryophytes, our liverworts and mosses, right? Their gametophyte is fairly large. You can actually hold it in your hand, right? The sporophyte is pretty small, right? We are actually, unfortunately, going to do something really confusing. This is just the way it is. In ferns and in seed plants, the sporophyte is growing larger and the gametophyte is becoming more microscopic. So it's kind of reversed for the rest of this, these groups, these divisions. So the sporophyte in ferns and in seed plants, trees and things, that's the giant leafy green part that you're going to be holding in your hand or seeing, right? The gametophyte is microscopic. You won't be able to see it as well. And in seed plants, the sporophyte would be like a giant tree, and the gametophyte is only a couple of cells large. So pollen grain, just a couple cells large, and the egg cells are really, really tiny. Okay. So again, here you can see in bryophytes, the sporophyte and the gametophyte, but now in ferns, your sporophyte's getting huge. In seed plants, your sporophyte is giant, could be as big as a sequoia tree, right? And gametophyte's just really small in both of those groups. So here we are. We are now moving from bryophytes into our lycophytes. And we are getting this new evolutionary trait called vascular tissue. So What's really cool is now these guys down here, they were gametophyte dominant. You mostly saw them in the gametophyte little grassy green stage. Now we're getting into sporophyte dominant. So the sporophyte, again, is the big leafy green part in our ferns and the rest of the tree. But that's what you primarily see them in. So tracheophytes, that just means that the plant has vascular tissue. And the two types of vascular tissue you're going to be responsible for knowing are xylem and phloem. I like to say that the xylem transports water and minerals, right, from the roots up, okay, to the rest of the plant. And the phloem flows down, right? So it's transporting sugars or photosynthetic products from the photosynthetic parts of the plant, like the leaves or the stems, down and all around to the rest of the plant, okay? So our first division of plants with vascular tissue that we're going to talk about are our Lycophytes. So our Lycophyta still produce spores, okay? But the most important part here is that they are going to be sporophyte dominant now. So you will primarily see them in this stage, okay, with their little spore-bearing structure up top. They do have some other types of structural support like lignin and cellulose, just helps them grow taller and stand upright. We do, like I mentioned before, have small gametophytes, okay? And they are independent. They do not need to grow out of the sporophyte or anything like that. The sperm is flagellated. We still need water to reproduce. And we do have what are called stomata for gas exchange. So, in photosynthesis, we're taking in carbon dioxide into our stomata and we're putting out oxygen, right? And we're also putting in water vapor, right, into our stomata during photosynthesis. So we also have some mycorrhizae, which is a little fungus that we talked about last week, helps plants grow and absorb nutrients a little better. But here you can see an open stomata. This is under a microscope, obviously, and then closed stomata right here. So open and closed for gas exchange. So our group, our order, Lycopodiales, so this division, Lycophyta, consists of two groups, okay? So Lycopodiales and Selaginales. So our first group that we're going to talk about, Lycopodiales. These guys have a sporophyte body, the leafy green body, that exhibits what's called dichotomous branching. It just means that, you start at a point and you're going to split into two parts. Like think of a forked road, right? Again, once you get to that next point, that next node, you're going to split into two again, and it keeps going. So we don't have true leaves. We have what are called microphylls. So these are just not quite true leaves yet. Our spores are produced in structures called strobili. So here is a strobilus right here. And we only have one type of spore, so it's homosporous. That's where that word comes from. And spores will become monoecious gametophytes. So much terminology here. Monoecious just means that both male and female parts are on the same plant, okay? So these guys do not have separate gametophytes like we did in the mosses and the liverworts. We just have, male and female parts on one structure, okay? So here is a Lycopodium strobilus. We have these little spore deposits here. So on an exam, you may be given this, and I might ask you, what is this? What are you looking at here? Well, these are spores, right? You would just need to know that this is a strobilus of some kind. You do not need to know that it's from Lycopodium. Just need to know that, strobilus of some kind, and it has some spores, spore deposits inside, okay? And again, these spores are housed inside of the sporangium, right? So this IUM ending, the suffix there, just means house, right? So it's a house for the spores. So this whole pocket here is called a sporangium. Inside, all the little dots are the spores, okay? Selaginelles. So selaginelles are spike mosses, right? So they exhibit dichotomous branching, and they have their little, their small leaves, their leaf-like structures in sets, okay? So you have a larger leaf set, and you have a smaller leaf set in the middle. So you will need to draw this. This is drawing number 14, I believe, in your lab exercises. If my numbers are off a little bit, don't mind that. Just go back and find about where you think this should go, but it should ask you to draw the leaf branching in selaginelles. So these are heterosporous, meaning we have different sizes and types of spores. The spores, just like in the Lycophyta, the Lycopodiales, they're going to produce in strobili as well. We have different types of spores, right? We have megasporangia, which are houses for our female gametophyte that has the egg. And then we have microsporangia, which house the male gametophyte with the sperm, okay? So if you ever see these words, I'll tell you right now, think micro, just like micro, if you've played Cards Against Humanity or whatever, think micro penis, right? I know it's ridiculous, but we're all adults here. It'll just help you remember. It's just a stupid thing, right? But mega would be the female part, okay? So micro, male, mega, female. You will never forget ever again for the rest of your life. You are welcome. So here is the alternation of generations for our Lycophyta, our two groups, Lycopodiales and selaginelles. If we're starting with spores are produced here, right? So we get these two different types of spores. They're going to fuse. The microspore, which produces the sperm, is going to fuse with the egg cells housed inside of the archegonia. They're going to find a way inside of that megaspore, fuse together, create a zygote. That zygote's going to turn into a sporophyte after a while, or embryo, then sporophyte, right? And then it's going to grow up, produce a strobilus with all of those spores again, and do this all over again in order to reproduce. We are out of our Lycophyta. We are into a new division, Monilophyta. So you may also hear this word Pteridophytes. This just means ferns, right? So our spore-producing vascular plants. So these guys are sporophyte dominant. They still require water in order to reproduce. Their sperm are flagellated still. We do have true leaves this time, and our true leaves, as opposed to microphylls, which were the not true leaves, we have what are called megaphylls, which are just larger true leaves, okay? So we have rhizomes. We also have some true roots in this division. So the rhizomes, root-like structures, and true roots. We can do asexual reproduction, either through things like the gemme cups or just breaking off a piece of our roots. And these guys are considered epiphytic, meaning that they climb. They can climb onto other trees and grow that way. They can grow out of water, or they can just grow on land, and they are terrestrial. So our first order that we're going to talk about, our first division, is Psilotales. These are our whisk ferns, and they have these specialized structure, really the only thing you need to know on this slide, specialized structure called a synagium. The synagium is basically 3 sporangia, or 3 houses for spores fused together, okay? And so all those little dots, all the little white dots are spores inside. They also exhibit dichotomous branching, just like in some of our other plants that we just talked about. And here you can see they're epiphytic. They grow onto other structures. So this, whisk fern's growing out of a tree, right? So here's our synagium. This is the drawing that you'll need to do. We have 3 sporangia fused together, okay? And inside, all those little dots are our spores. Remember, synagium is actually diploid. The spores itself are haploid, okay? Next order is Equisitales. These guys are homosporous. They only have one type of spore. What you need to know is that their strobili here, where the spores are housed, look like this, all right? They have reduced leaves, which wrap around the stem just like that. Their spores have what are called elaters, and it just helps them when they're dispersed out of the strobilus, they can attach onto things a little easier, more easily, right? So here is one little bract called a sporangiophore pulled out, and you can see that. You can see this single group here is a sporangium, and inside are all the spores that are the darker dots, okay? Our next group, which are our traditional ferns that we're used to seeing, like all around campus or in the woods, these are polypodiales, okay? So these are leptosporangiate ferns. They only have one type of spore. They have roots, fronds. So our structure here, this whole thing is called a frond, okay? This entire thing. We have these little leaflets, okay? These are little blades, right? Blades of leaves, leaflets. But this whole thing is one leaf, okay? And we have this here, okay? We have this sporangium. There are lots of dots on the back of here, and I'll show you more what they look like in a second, but we have these structures called sporangia still that house our spores, right? So on this slide, you can see it a bit better. If you turn a fern frond over to the back side, you'll see these dots on the back. Each dot that you can see is called a sorus, okay? So singular, it's sorus. If you're looking at a bunch of dots, it's called sori, okay? So if you isolate one of these sori, so into one sorus, right? You'll notice they're made up of a lot of these sporangia, okay? So inside of there, lots of sporangia, and the spores, there are just thousands and thousands of spores on the back of a fern frond, okay? So what's gonna happen? The way that spores are, dispersed, they have this structure called an annulus, right? An annulus, it's going to dry up, okay? If we were in the lab, actually we would dry it up with alcohol and we could see them pop open, but that annulus is gonna dry up and then the spores will fling out, okay? And then they are dispersed and able to go germinate into a nice little gametophyte that's gonna grow into a sporophyte, right? After a while. So here is our alternation of generations for our ferns. Like I mentioned before, spore germinates into a young gametophyte, which is monoecious, meaning it has both male and female parts on it. We have the antheridium, which produces the sperm, and then we have the archegonium, which produces the egg. They're going to fuse together via fertilization into a diploid zygote. Zygote's going to grow up into a sporophyte, which is it looks like it's growing out of the gametophyte, but I'll tell you, it's just independent, and that gametophyte shrivels up and dies, okay? It does not need the sporophyte at all. And then it's going to grow into a nice mature sporophyte, which produces spores through meiosis, and the cycle goes on, okay? So if you're looking at a gametophyte, you'll notice that it looks like Lady Gaga, pretty much, one of her many outfits. A scientist actually named one of the genes in the gametophyte after her. It's called the Gaga gene now. But the archegonium, where we get our eggs from, is housed towards the dip right up here on this heart-shaped structure. And then all of the sperm cell structures, which are called antheridia, those house the sperm, are going to be towards the bottom, okay, on the sides. All right, so eggs up here, sperm cells down here. So what you need to know for our practical exam coming up, you need to be able to describe the alternation of generations. You'll need to know a little bit about each of those groups, primarily what I told you to remember, right? Know the vocab words that I bolded and colored, right? And maybe said a bunch of times, and be able to identify those plants. We're going to give you the same exact pictures that you saw in this PowerPoint on the exam, and you'll just need to be able to identify them, look at them, and then say, oh, that is a fern, right? Or, oh, that is a bryophyte, that's a moss, okay? So now you're going to go to your lab activity. This is what your lab looks like this week. go through. You can answer all of these questions. I don't think that there is a question that you can't answer. So this week, some other useful terms, right, at the end of the PowerPoint. So these are things you could add to a Quizlet or make flashcards out of. If I ever ask you for the ploidy, I'm asking whether it's haploid or diploid, all that good stuff. For next week, make sure to submit your group article. So speaking of which, if you go to if you're on Canvas, right, and you go to the home page and you scroll down to Species Spotlight topic sign up, right? So this is for my Monday class. If you did not sign up, I've put your name somewhere. Notice how some of these groups have 5 people or just one group of 5. I have a feeling that someone is going to dip out. If they don't, you'll just have a group of 5, and you'll need to contact each other. But all these are finalized groups. When you go to turn in your assignment, so notice how under assignments, you'll have a section now called Species Spotlight Submissions. To you, you can only see this one submission. I only need one person from your group to turn this in. It automatically, I've set you up in groups already, according to what was on that page. So if you turn it in, you've turned it in for your entire group. So you're good to go. So you'll upload whatever article you find as a PDF here. Remember, before you move forward with this project, I have to approve of your article. If for some reason you choose an article that doesn't work for this project, you'll have to find a new one. Okay, so be careful. Make sure you know that. Make sure you're constantly talking to your groups, whether email, start a group chat, start a Snapchat group, whatever you need to do in order to stay connected. Okay. Lab 4 submission. Every week, I'm going to write exactly what you need to do in order to complete this lab, okay? So this week, you need to upload manual pages 21 to 28. Don't lose that page 28 because we're going to be filling it in next week as well, okay? So make sure you upload all that and you should be good to go. Talk to your groups. All right, if you have any questions, just reach out, contact me. I'm happy to help you, okay? Come to office hours. If I am not in the office hour room for any reason, just send me an email. But I get an email every time someone tries to join the session. So just hang out for just a moment and I'll be right on as soon as I get that email. All right, so have a great week.